So let's continue to talk about the eye. Remember that the wavelength that is visible to the human eye is between 400 and 700 nanometers. So if you look at the scale, you can see that UV light, X-rays, infrared, microwaves, any of this is visible to our eyes, right? There is a little portion of UV and infrared that we can see, but not the smallest wavelengths and the bigger wavelengths from infrared. Those we cannot see. One thing that is important here is that when you think about the visible light possibility, you look at this, how do you call this? My God. Spectrum. And there are different wavelengths that are can be seen by different photoreceptors. So for example, blue cones can see only wavelengths that are around 420 nanometers. Rods are here, you see, in between blue, red, and green, green and red. Greens have a bigger wavelength and red even bigger according to the spectrum of light, right? And then you say, okay, but based on this, we would only be able to see blue, green, and red. So how do we see the other colors? We see the other colors because the eye can perceive a mixture, can receive that light that is, for example, a shade of green, and the mixture of cones will allow you to see that shade, that different tone, that different color, okay? On the other side, rods, because they are very sensitive to light and you can use them to see in the dark, you don't usually see images when you look with your rods that are a specific color. We don't see color in the dark, right? So rods don't really see color, and they're just one specific wavelength, which is the peak is in 500 nanometers. One more thing I wanted to mention, focus. So when we're focusing on an object, remember that refraction is gonna happen in the cornea, in the lens, and once the image hits the retina here, your image is going to be upside down and inverted, okay? And then you put back in the regular, you don't see the world upside down because this repositioning of the image is what happens in your occipital cortex, okay? The vision cortex. So when you're looking at something at a distance, so let's say this is an image that you're looking at from far, you are flattening the lens, and that is happening because this muscle, the ciliary muscle, is pulling the lens to flatten. The opposite happens when you're seeing from close. You see how now the lens is much more round if you compare to this, right? See the difference? So here we're seeing from close and you bulge the lenses. There are three conditions that are important for us to understand here. One is the myopia. What happens is that the eyeball in the myopia is too long. So if this would be the perfect eyeball, when the eye is too long, instead of your image focusing and in the retina, you will focus on this point, and this point is not the retina. So by the time the image gets to the retina, whatever you're seeing from close gets distorted. Sorry, whatever you're seeing from far gets distorted. You are nearsighted when you're myopic, meaning you don't have a good far vision, okay? And you can correct this with concave lenses. The concave lenses are going to help to focus your image in the retina of this long eye, right? What's the issue with hyperopia? In hyperopia, you have the opposite problem. The eye is too short. So instead of the image from something you're seeing from close, so the problem is with near vision, focus in your retina is going to focus in the back, like you would focus behind your retina. So what happens in the retina is not a clear object, is a blurred object of something that you're seeing from close. You correct it by using convex lenses, and these lenses will adjust for the focal point to be formed in the short eye retina. Okay, so now let's just look at the differences between rods and cones. Like I said, rods are important for non-colored vision, high sensitivity, so they function in dim light. Cones, color vision, 3 different visual pigments that can be combined and you can see shades of different colors. And low sensitivity, function in bright light only. The acuity for rods is worse than for cones. So cones have high acuity because one cone connects to one ganglion cell in the fovea. And rods, you have many rods connecting to one ganglion cell. You have 20 rods for every cone in your eye. So less cones, but they're more accurate, right? And you have mostly rods in the peripheral retina and cones in the central retina. The part that is right in the center, right of the back of your eyeball, that's where you have mostly cones. What is color blindness? When you miss one or more cone pigments, so specific types of cone cells, cone photoreceptors, are missing, and then you cannot see some colors. More common in men. Now let's talk a little bit about adaptation. So adaptation is when you are in either a super bright place. Oops, I'm trying to make this fit my screen. You're either in a very bright place and then you go to a dark place, or the other way. So we're going to talk about light and dark adaptation. So remember that you need vitamin A to make retinol. And retinol is a molecule that absorbs light. And when you combine retinol with opsins, which are proteins that are present in the photoreceptors, you form the visual pigments. So there are different visual pigments. For example, there are different ones that you can find in the cones, and they are used for you to be able to see it. So the cone opsins are very specific for each one of the colors: red, green, and blue. The rods opsins are called rhodopsins. So rhodopsin is a molecule that allows for adaptation from light to dark, from dark to light. So rhodopsin is also made of vitamin A, so you need vitamin A to be able to make it. And in the presence of light, rhodopsin is broken down. So what happens? When you are moving from a dark place to a bright light place, your rhodopsin breaks down. Anytime there is light, rhodopsin breaks into retinol and opsin. That breakage is going to cause a glare in your eye. That's why you go like, whoa, that looks so bright, even though later on you don't think it's that bright, right? So you need to wait for that period of breaking down the rhodopsin to happen so you can adapt the vision to light. That usually takes from 5 to 10 minutes for light adaptation to happen. When you talk about dark adaptation, from bright light to darkness, then it's the other way. You have to wait, you get in the dark, you can't see anything, because what do you need in the dark? You need rhodopsin in your rods, right? Remember, you use the rods to see in the dark? So rhodopsin needs to be rebuilt, resynthesized for you to be able to see. The problem is that for the regeneration of rhodopsin to happen, it takes usually 20 to 30 minutes. That's why dark adaptation is slower than light adaptation, okay? Okay, let's change the subject. Let's talk about sense of smell and sense of taste. Why are we going to talk about them together? Because they are together. You have chemoreceptors, so they identify chemicals, right? That are either dissolved in aqueous solution or in the air for you to be able to smell and taste. So when you smell, remember that you have the olfactory epithelium here, which is your organ of smell. Let me get a different color. And the olfactory epithelium has the nerve endings just going through the perpendicular plate of your ethmoid bone, and they hang in your nasal cavity. Because you have the nasal conchae, the air makes a turbulence. The nasal conchae are all these little invaginations in your nasal cavity. The air goes through a turbulence and is able to reach the olfactory epithelium that is all in the top here before the air goes down. That's how you're able to smell. There are 100 different odorants, and we have 400 smell genes active in our nose. When we think about the taste buds, so they're located in your tongue, and they're also located in your mouth, in your soft palate, cheek, pharynx, and epiglottis. So there are some also there. I'm not going to ask you about the 3 different types of papillae, which are where the taste buds are, but this is the tongue, and it's just showing you how the taste buds are distributed all over your tongue. You have, contrary to common knowledge, I think, some people say, oh, you use the front of your mouth for a specific type of taste and the back of your mouth for another type of taste. What the book suggests and explains is that all these different papilla are able to taste all the different tastes. So when you look at this is an enlarged papilla. That's where the taste buds are. Each one of these little blue things here are taste buds. Do you understand why if the chemical, let's say you put a spoon of sugar or salt in your tongue, if you didn't have saliva, you would never be able to taste it. Why? Because the saliva is the liquid that will bring that solubilized sugar or salt to this little invagination where the taste buds are. So without water, you don't taste. Okay, that's why it's so important to have saliva for the sense of taste. Remember that taste, 80% of your taste, I think it's 80%, comes from smell. So when you can smell, these neurons are connected. And when you can't smell, you also taste much less. Okay? That's why they usually talk about loss, if a disease causes loss of the sense of smell, it also causes loss of the sense of taste. When you have a cold or the flu, you are not going to be able to smell and taste much because everything is inflamed, right? So this is a taste bud. One of those little blue things that I showed you in the previous picture is actually this. And this is the taste spore. You see? So the chemical comes through here. And then you see where the neurons from the gustatory nerve come from? So all the way here. Remember, the hypoglossal nerve is the one that sends fibers to the taste buds. So the free endings here are the ones that are going to detect the chemicals, okay? There are epithelial cells inside the taste buds that allow for this to happen. Taste, you have all these different types of tastes that you can sense with your taste buds, okay? So gustatory pathway, glossopharyngeal, vagus nerve, and facial nerve. Sorry, I said hypoglossal is actually glossopharyngeal. Hypoglossal is more for tongue movement. And remember that the gustatory cortex is in the insula, is the part inside. If you open your temporal lobe, your temporal cortex, if you remove that, then you could see the insula where the gustatory cortex is, which is right here, okay? So neurons from vagus, glossopharyngeal, and facial nerves travel through the pons, thalamus, and then gustatory cortex. And it induces salivation, gastric juice production, gagging, and vomiting, all also controlled by these nerves, okay? Okay, I think I'm gonna stop here and make a new video for you about the ear. Bye-bye.